Linda, we're now at the cloth lab. Am I pronouncing that right?
Yes, cloth.
Okay, what is it that you do here?
Dr. Clough works on several different aspects of soybean. One of the technologies he uses is called microarray technology. What that allows him to do is look at gene expression over time, or gene expression at a certain point in a or certain event that's happening in the soybean. For example, if we want to know what genes are expressed, say what resistance genes might be expressed when a soybean plant is being attacked by a pathogen, microarray technology lets us identify those genes that are being expressed, which may be resistance genes that we want to know about when the pathogen is attacking the plant.
So the trick is to isolate those genes.
Yes, and that can be a tricky business, and microarray technology lets us get at that business so we can begin to understand the genetics of resistance reactions.
Okay, let's go ahead and get into the lab then.
So what you see is a lot of different equipment. It's hard to be able to explain what all of this different equipment does. So you can see that it kind of looks like
it looks busy.
It looks busy. Yes, it looks very busy.
And there's nothing wrong with that.
No. Usually during the day you have a lot of activity in here. They heard we were coming, so they thought maybe they'd take a break.
Well, we should say we're close to the end of the day. The people who work here, are they working as students as part of the university, or are they employees of the lab? How does that work?
So in most labs, and in Steve's lab, what we're going to have as staff members would be students, master's students, PhD students, or postdocs, those individuals that have finished a PhD, and before they look into jobs available in the industry, they might do one or two postdoc positions that gives them experience working on a certain research project, and maybe allows them to write one or two more publications before they're out looking for a job, and it helps them with their competitiveness on the job. And then also you have research specialists that can help in a lab. So we've got students, masters and PhD, postdocs, and research specialists.
And what kind of things will they be working on here?
This is the USDA soybean lab. So they're going to be working on soybeans. They might be working on gene expression for soybean development, gene expression for disease reaction, for say when the soybean begins to produce a pod and we want to know what genes contribute to oil production or protein production, we might be looking at gene expression during pod formation.
Anything else you'd like to show us here?
Yes. There's another project that's gone on here at the University of Illinois. It's a very unique project. University of Illinois holds the patent for this particular process. And I might explain that a little bit before I show you some really interesting little soybean plants. A little earlier we were talking about where do you go find new genes for resistance? Where do you find them? And one of the sources has been, say, a remotely related cousin of soybean, glycine tomentella. So there's an investigator, Dr. Ram Singh, that's worked his whole life on breeding or moving genes from tomentella into glycine max, which is soybean. There's a little problem with biology in that if you try to cross these two plants, then you cannot produce fertile offspring. So it's like, to borrow an animal analogy, it's like crossing a horse with a donkey, and you have a mule, but a mule and a mule cannot breed. So when we cross glycine max with glycine tomentella, we get offspring that are an evolutionary dead end. They cannot produce other offsprings or viable seed. The seed are sterile or the pods abort. Well, if you have some very interesting traits in glycine tomentella from Australia and you want to move them into glycine max, Dr. Ram Singh has made that possible. He's overcome some biological barriers. And in this process that he's patented, one of the one of the processes, or one of the roads, I guess, to that particular success is culturing plants in growth medium. And I'd like to show you some of these plants too. So let's go over here. So what he has to do is a long process. It takes 3 to 5 years to accomplish. And during this rather long process, he produces soybean plants in culture that are very tiny. As you can see
show
here.
that just about.
So to get to this point,
Maybe a little bit lower there. I think we can get less glare that way. Okay.
to get to this point, what Dr. Singh has done is made crosses between Tomentella and Glycine max, and the pods want to abort, and that's the end of the process. He rescues the embryo out of the pods that want to die at a certain interval. There's a few days in which he can rescue the embryo, puts it into culture, and produces through tissue culture small masses of cells. And then he treats these cells with certain plant hormones and chemicals and which allows them then to produce leaves, but no roots. And at this point too, he has to modify the chromosome number so that the plants that will eventually be produced are fertile. Because the problem is we've got the wrong chromosome number here. So he can modify the chromosome number with chemicals and then produce a soybean plant that still has the wrong chromosome number, but grow this up and then put it in another growth medium that has a rooting hormone, root the plants, grow them in the greenhouse, and then begin back crossing them to the Glycine max parent that was originally used. And with each back cross, the chromosome number adjusts, and you'll eventually get from 118 chromosomes, which is what we have here, to the 40 chromosomes that Glycine max has. Along the way, he's dragging chromosomes from the Tomentella genome into the Glycine max genome. And any of those traits that are desirable traits, which may be resistance to soybean rust or soybean cyst nematode or sclerotinia stem rot, or they may be yield genes, they may be drought resistant genes. And we don't know yet. So we're at a point where we're going to begin to screen the varieties, or the germplasm actually that's in the greenhouse, for certain economically desirable traits.
His job takes both patience and persistence, doesn't it?
My goodness, it has. It's been a lifelong challenge for Ram, and it's great to know that, you know, at the end of his career, and when working with this particular cross, that he's been able to patent the process, and we're now understanding what a treasure trove of traits that we may have in his life's work.
So this particular process is something that's unique to the University of Illinois?
Yes, it is. Yes, it is.
Wow.
Yes.
Well, it is something to be proud about and have a lot of satisfaction about as well. For him to be able to do this, and a lot of the work here, I would imagine you need to have access to a huge variety of soybean seeds. Do you have a seed bank here as well?
Yes. Yes, we do. The United States Department of Agriculture soybean germplasm collection is located here on the University of Illinois campus at the South Farms. And there are, at last count, about 21,000 plant introductions in that collection. It's a wonderful resource. We haven't screened all the varieties or all the PIs that are in that particular germplasm collection. So that's one of the efforts that's ongoing, is to screen that germplasm for economically important traits, and also increase that germplasm too, which there are efforts between university scientists and folks in other countries in movement of germplasm between the countries, and information between the countries too.
Does all of this make the University of Illinois, this institution, the leading lab in the world?
There are several leading labs in the world, and University of Illinois is definitely among them when it comes to soybean research.
The leading lab in the United States for soybean research?
I'd like to say that, yes. We'll claim that honor right now.
And there aren't people in Iowa or Indiana or other places that would disagree with you?
We all do good research, and we exchange information, and we have some wonderful projects with soybean laboratories in other states.
I think you're ready to be a diplomat. Thank you very much, Linda. Anything else here?
No, thank you very much for the opportunity.
Okay.